So please give us a talk.
Thank you so much. Thanks a lot for the invitation. Thanks to the organizers. And I will have some trouble with this presentation because my computer wants to get updated, but I will try to do my best. There was a fantastic talk right before, and my presentation will relate in many different ways, and so I'll try to make the connections. But it was fantastic. It will help for my presentation, so it will stay on the same theme. So I work at the New York Genome Center, and as I already invited some of you, we have a very nice building down 6th Avenue, and so if any of you wants to visit tomorrow, we can definitely organize something. But I need to mention, because we're on 57th Street, so when I joined the New York Genome Center, our office, we had a very small office that was like about half the size of this room, was also on 57th Street and Broadway, so really next by. And so in 2013, we moved to this nice building in Soho on 6th Avenue and Canal Street. And so that was really created by a network of institutions, university and hospitals in New York. The goal was really to establish a place for large-scale genomics project in New York City. So we are focusing on cancer, neuropsychiatric disease, and neurodegenerative disease. And you can see here the logo of all the institutions that compose our network, all of them located in the New York region. So we're primarily a genome center, so we have sequencers. For about 10 years, we entirely focused on Illumina sequencing, so short read sequencing, in which we can do RNA sequencing, we can do exome sequencing, we can do whole genome sequencing. But more recently, we also expanded the technology that we use to include long read technology. One instrument is mentioned here, Oxford Nanopore technology, and the instrument is called Prometheon, where we can sequence reads that are much, much longer, and that's complementary to what we do with Illumina. There is also this large black box in the middle of my slide. That's an instrument from a company called Ultima Genomics that recently became public, where we can do whole genome sequencing for a cheaper cost than Illumina. And so that's an alternative technology that's cheaper, that has pros and cons, that has different error models, and we can use that for various applications. And then what we do also at the New York Genome Center is to develop new protocol, in particular for single-cell genomics, both single-cell whole genome, single-cell RNA-seq, multimodal single-cell genomics, combining RNA and protein measures, and the new wave of technologies around spatial transcriptomics, where instead of having one sample that we mash together and we sequence, we can identify what are the different expression pattern, for instance, on a slide or on a particular tissue, and we can study very precisely the expression of the cells around the tumors and the one inside the tumors, the one that are at the middle of the tumor or at the periphery of the tumors. All of that type of information can be revealed by spatial transcriptomics approach. So here is my technical slide. I promised him to one of you. That's the pipeline that we use for whole genome sequencing. So we sequence both the tumor samples and the normal samples, typically from a blood sample. And the goal is to identify the differences between the two, right? What are the mutations that are acquired in the tumor that were not found in the original cell and the original normal genome of the individual? And so we apply a combination of tools. So we take the Illumina short reads, we align them to the reference genome, and then we have a combination of tools to identify all of the different categories of variants that can occur in the tumor. So short single nucleotide variants, small insertion and deletion, structural variant, translocations, whole genome duplications, all of those variants that can occur in different cancer contexts, we try to detect them with our pipeline that we published a couple of years ago that's accessible, that can run on a local computer or on the cloud infrastructure, and we applied that to various different cancer types, different projects. I listed some of them here. We also have a pipeline for RNA sequencing, so it's slightly different type of information, and we infer different type of information, the presence of gene fusions, the expression of the genes, and we can study the differential expression and so on and so forth. And there's really a large list of downstream analyses that we can do when we have this type of information. And so an example that we recently published is a project that we've done with colleagues at Weill Cornell, where we study brain metastasis coming from prostate cancer. So many cancer have metastases in different locations. Frequently, they go to the brain. I'm sorry about that. But in prostate cancer, it's fairly rare. And so we obtained some samples, and we did whole genome sequencing, both of the normal genome, the primary tumor, and the brain metastases. And we tried to study what is predicting why do some patients have brain metastases. And so we assemble all of this information, all of the genes that were mutated, all of the category of structural variants that we observe, and we really try to illuminate and try to understand a little bit more why do some patients have brain metastases. And you know, at the end of the day, that's a small study. We don't have a lot of samples, and we didn't find like a clear pattern. But it's very useful data that we hope that someday will become very useful for the patients. Another type of project, also with colleagues at Weill Cornell, is to work with rapid autopsy. And so there are programs that patients agree to donate their entire body to science at the time of their death, and the autopsy program can collect different samples. And so here we have primary tumors that were collected during the evolution of the tumor, as well as multiple autopsy samples, metastatic samples obtained at the time of death. And so we can do whole genome sequencing on all of those samples. Here we have 12 whole genome, and then we have 5 plasma whole genomes that were collected during the evolution of the tumor, and we can relate all of those patterns. So what you see here is a phylogenetic reconstruction of the variants that occur early in the disease that were common to all of those samples, and the ones that are more unique to the different samples that we observe. What you also can notice is that we color those samples in pink and green because they correspond to 2 different histology. It's a very rare case of a patient where we collected samples that had different histology, and the goal of the genomic work here is to understand what switch occurred during the evolution of the tumor that explained that some of the samples completely changed their behavior and really showed differently in the pathology slide. So that's an ongoing work that we're trying to finish soon. And the final example I want to show is a work on bladder cancer, where again we used rapid autopsy samples, and we studied the relation between all of the samples, and for which what you see on this phylogenetic tree is the small nodes, the small circles, are colored by the somatic signatures that we just talked about. And so here what we see are in the early mutations that are occurring early in the evolution of the tumor, most of the mutations are corresponding to ApoBec, which is one of the mutational processes that occur in many different cancer. We see that early on in the evolution of the tumor. And as soon as the patient is diagnosed and is given chemotherapy, we start to see a lot of subclonal mutations corresponding to the chemotherapy pattern of signature. So it's specific signatures that is associated with chemotherapy that we see later on in the evolution of the tumor. And so here from all of those samples from that particular patients, we see both the early mutational process and the late mutational process. Another thing that we studied in this particular cohort is the existence of an extrachromosomal DNA that amplify a particular gene called cyclin D1. And so that's also a pattern that we observe and that we studied quite extensively in this cohort, including by doing long-read Oxford nanopore sequencing on this particular extrachromosomal DNA, and we hope that's going to be published soon. So just final slide on the type of work we do on liquid biopsy, and in particular for minimal residual disease. And so that's a type of experiments where we can take a blood sample, typically after a surgery or after an operation for patients that was treated for cancer, and we can use whole genome sequencing to measure the recurrence of the disease and to measure how much tumor fraction is present in the blood of that particular patient. And we do that with whole genome sequencing in different fashion or different AI models or different deep learning models that apply to the data to try to infer what are the signals that tells that cancer is still present and potentially might grow again for these particular patients. So that's the type of work, and that's fairly unrelated to cancer health disparity. But what problem we have in cancer genomics is that we use a lot some database that are shared by all of the scientific community, and those databases are extremely non-representative of the large patient population that we want to work with. So here are two examples of known, very well-established public database. One of them is TCGA, so the Cancer Genome Atlas, and this pie chart corresponds to the self-declaration of the patients. So in TCGA, about 80% of the patients are self-declared white or European, okay? And so the rest is like a small minority of the cohort are Black or African American or Asian, okay? That study was done in the US, but still it does not represent the whole racial or ethnic diversity that we have in this country. The small pie chart that you have in the middle here correspond to the same information but using genetic ancestry, and I will describe it in a little bit more detail how we do that, but you see that the two informations are quite correlated. On the right side of the slide, we have the AACR Gini, so it's a larger cohort, larger project with more samples. It's a different type of experiment, but still we have a very large representation problem where most of the data that we have in those databases are from a very small subset of the population. And so when we try to aggregate all of the information and try to build model to learn from those information and then apply that to different patients, that might not work because the patients are not present in the database that we started with. And so really our goal is to try to diversify the database and try to understand, to make the database more representative. One very clear example of the change that we can have different between population and that will justify what I will present after is the example of EGFR mutations in lung cancer. And so lung cancer is the most common cancer, the most deadly cancer around the world. It's heavily studied. EGFR is a very known driver mutations. And so around the world, people have studied how many of the patients with lung cancer have a mutation in EGFR. And we see that the proportion of the patients is about 10% in Africa, 10% in Europe. And as we go along the human migration, we see that it goes to 23% in India and 50% in East Asia, and then goes to the US where it's again 12 or 10%. And in Latin America and South America, it goes again to 33%, 50% in Peru, and 14% in Argentina. So there is a lot of variation, and it's really based on large numbers. We don't really understand that variations. It's not just due to diet, it's not just due to environment. There is very likely a genetic cause that can relate to these very, very large variations between the different populations. And so we're in New York, we're the New York Genome Center, and here is a map of New York colored by the census. And so you see those categories here are what people ask at the census. Is it Black, Hispanic, Asian, white, or others? And we colored the map of New York with what the neighborhood is mostly composed of. And so you can see in Manhattan, we have a lot of white populations. You can observe these red dots corresponding to Chinatown in the south of the island of Manhattan. On the north, we have neighborhoods that are predominantly Black, predominantly Hispanic. And if you go to Queens, if you go to Brooklyn, you have different compositions, okay? So we have a lot of diversity. We also have a lot of segregation between the neighborhood. And that relates definitely to the disparity that we observe. And so with this situation, we build a project that we call Polyethnic 1000. And here is a summary of the pilot phase for which we published an essay describing it, where we collected 160 tumor samples from 13 institutions. We did whole exome sequencing and RNA sequencing. It was various cancer type, but it was really just a demonstration of what we could do. And this bar plot here correspond to the genetic ancestry. And so the top bar here correspond to the self-declaration. So you have a lot of patients that were self-declared, entered at the hospital as Black or African American, and then some were Asians. And the middle plot here correspond to the percentage of their genome that we associate with the African continent in orange, with the European continent in green, and with the Asian continent in red, okay? So most of the patients that were self-declared Asian had predominantly their genome associated with the Asian continent. A large proportion of the patients that were Black or African American had a variable proportion of their genome corresponding to the African continent, and so on and so forth. And you can, the level below correspond to the different population. So we use a project called 1000 Genome Project that collected samples from 25 different populations around the world. And so you can see that in the African continent, you have people with most of their genome associated with the African continent, the type or the reference population they associate with is quite variable. And so you have a lot of genetic diversity in Africa. We can measure how much of their genome corresponds to the African continent, but then they can come from different places in Africa. And there is a lot of genetic diversity between those places, right? And so that's really the level of information that we want to work with, is not so much the self-declaration, because those are broad categories that do not really inform on the genetic markers that will be present in those patients. But if we can get a little bit more precise, we can quantify their ancestry, where did their ancestors live, and really have a proxy for the genetic variants that are likely to be present in those patients. So that's the way we work with genetic ancestry estimations. And so here, that's a different way to represent, but the first thing we do when we do that is to look at markers of genetic ancestry and run a principal component analysis that is really a standard method in population genetics, where we can look at the variants that vary between samples, and then run some clustering analysis to quantify the portion of the genome of each individual patient corresponding to those different categories, to those different continents. Our goal is not to put patients into categories, but it's to measure their association or their correlation with particular reference population that we use, okay? And so the second phase of the project that I want to describe now is we ask a lot of people in New York, in their particular cancer of interest, how would they use the network and the infrastructure that we built. We assembled a review board of fantastic cancer scientists that were not in New York, that were not associated with any of our partners, and that help us to rank the different projects that we receive. We had a large support from various industry, from philanthropic donors, and from universities that allow us to do 1,000 retrospective and prospective samples on which we do tumor normal whole genome sequencing and tumor RNA-seq. And so the project that we're doing are listed here on those different cancer types. The institutions are listed, and the picture of the investigators are represented here. And so currently we are getting close to our number of 1,000. And here is the map of the proportion of the genetic ancestry estimations. And so you see from the left, the bladder cancer, breast cancer, colon cancer, 2 projects on endometrial cancer, lung cancer, multiple myeloma, pancreatic, and prostate. So the first thing you see is that we have a lot of orange. So remember, orange corresponds in our mapping to the African continent. We have a lot of patients that are self-declared as Black or African American that have a large African ancestry. The 2 exceptions I want to point out is the one on the left, bladder cancer. One reason for that is that we specifically focused on a cohort coming from Egypt, and Egypt being the place in the world where bladder cancer is the most prevalent, in large part because of a water parasite that exists in Egypt and in neighboring countries that seem to cause bladder cancer, and we're specifically studying that cohort. The other exception is the project in the middle, the lung cancer project, and I will describe that in a bit more detail right after. Similarly, we have here the summary of all the mutational signatures. So as was described, we can take all of the somatic variants that we identify in our cohort and decompose them into the mutational signatures. And so the key is located on the right. I'm sure we can see SBS 16 here, but I don't think it was present in any of the cancer that we looked in details. But so you have 3 tracks corresponding to the SBS, so the single base substitutions, the DBS, so when 2 bases are mutated, or the ID, which are the insertion and deletion. And for each of those type of variants, we have different patterns that can correspond to different mutational process, right? So one of them being aging, one of them being smoking, ApoBec. We have various patterns that are known to be associated with specific phenomena that we understand, and then there are some where we don't understand, we don't know what's going on. And so our goal here will be a little bit similar to what was presented before, identify how the mutational processes in our cohort might differ from what has been observed for the same cancer type in other population previously. Those plots are sorted by the red bars at the bottom, corresponding to SBS1, so the aging signatures. And we can see that the effect of that signature is different between the different population, more present in the prostate cohort than, for instance, in the breast cohort or the bladder cohort with our type of analysis. So really mostly here what we see, and you have to trust me on that, we mostly see the signatures that we expect for those particular cancer type, right? We know that we will have ApoBec in bladder cancer, we know that we'll have smoking signatures in lung cancer, and so on and so forth, but there are novel patterns that we start to observe and that we'll need to validate and replicate and really make sense of. So I promised I would focus a little bit more on the lung cancer, and so what's unique in this project compared to the other in the Polyethnic 1000 project is that it's the only one where we focused on the East Asian community, in particular in New York. And so we looked at pre-invasive and invasive adenocarcinoma, so really early cancer, and we have the cohort is half of it is from white patients, half of it is from East Asian patients, all collected in New York. And you see that if we look at the somatic signatures sorted this time by SBS4, the smoking signatures, we see that about half of the cohort has those orange bars at the bottom that correspond to SBS4, as well as a DBS and an ID signatures that are also known to be associated with smoking. And so that's represented here, ID3, DBS2, and SBS4. On the right side, you have the white patients. On the left side, you have the Asian patients. There is no obvious difference in the proportion of the samples that have those signatures, but there might be some other patterns that we are looking at in more details right now that might differ between those two. One of the big problems we have for this cohort, if you look at the metadata that we represented at the bottom of the plot, you have the gender information and you have the smoking status information. And as you can expect, smoking status, when the patient is a smoker, you observe those smoking signatures, SBS4, DBS2, and ID3. But as can also be expected, most of those patients that are smokers are men. And so we have a confounding factors of difference in sex, smoking habit, and the type of smoking signatures that we observe in this cohort. So again, we'll look in more detail, and there will definitely be more information about this. If I go back to my example of EGFR frequency, we here represented the type of mutations in EGFR in the two categories. So at the top are the Asians, the bottoms are the white, and you see that we have a lot more EGFR mutations in the Asian cohorts. So those people all lived in New York, and it might be different places in New York, and that might be a different environmental exposure. But if we take into consideration their smoking habit, the fact that they both live in New York, we still have a lot more EGFR mutations in the Asian cohort compared to the white cohort for those small numbers. Here is the representation of the different genes that we observe. So EGFR being the most mutated, and then we have the frequency of mutations in the Asians versus the European cohort, and so we are really trying to make sense of those type of information. But so here is a representation of all of the different mutations in the main genes that we have in that cohort. I will maybe go a little bit quicker, but another cohort of interest is the one focused on colorectal cancer that we are studying with partners at Cold Spring Harbor and on Long Island. And so here I represent the genetic ancestry. Most of the cohort is patients of African ancestry. And so if we look at the signatures, and this time we had at the top the tumor mutation burden, so the total number of mutations that were detected in those whole genome, we see that we have a large cohort that we would that associate with DNA mismatch repair. And so those patients have more mutations, and those mutations are corresponding to ID2, DBS7, SBS44, 26, and SBS6. All of those signatures are known to be associated with mismatch repair, and so we're now looking at either the germline mutations or the somatic mutations to see how what are the relation, what are the explanations for why those patients accumulate more mutations. And so another thing we can do is take the RNA-seq and look at immune deconvolution. And so when you take the RNA-seq of an entire sample, you have the expression of the gene in the tumor, but you also have the cells that are surrounding the tumor, and you can try to infer what are the proportion of the different immune cells that are surrounding the tumor, and does that vary in the cohort? And so we see here that if we sort by the amount of T cells, it's quite variable. There are some samples in which we have 10% of the immune compartment is composed of T cells, and we have some where it's 50%. And so that might also relate to the type of mutations. If the patients have a lot more mutations, that might be what we call a hot tumor with a lot more active immune infiltrations and immune reaction to the tumor. And so that's another type of analysis that we can carry and we can lead. Okay, so what we need now for this polyethnic 1000 project, finish the sample collection, and that's done. Continue to analyze the genomics and the clinical information. We will share all of the data, and we have started to share the lung cancer cohort on ICGC Argo, and so that will progressively be available for the scientific community. And we hope to publish multiple P1000 papers. One thing for those of you who are really in the weeds of computational biology, and that I'm happy to talk to, is the idea of a pangenome analysis. And so what we do is always aligning the reads to the reference population, so what is considered as the consensus sequence across the entire population. But we know that's not a faithful representation of the diversity, and we now have methods to use a much more complex representation that contains a lot more information, and that might be really beneficial for analysis, especially for patients that are different from the standard or from the patients that we have studied so far. And so here for a cohort like polyethnic 1000, there might be an advantage at using a reference that's a lot more diverse, that's a lot more complex than using the linear reference that we use as a consensus. And so we are trying to develop tools that can do this type of analysis, this type of detection of somatic variant in a pangenome representation. And finally, what's really next for this type of project, once P1000 will be finished, is the project that we announced 2 weeks ago as part of the Cancer Grand Challenge as well, and that we call SAMBI, where we will focus this time on 3 cancer types: breast cancer, prostate cancer, and pancreatic cancer. We will collect samples in 3 continents, in the UK, in Europe, in Africa, in about 10 different countries in Africa, and in the US. And we will not just do the genomics, but we will also look at social determinant of health, exposomics, genomics, and immune profiling. And so that's a new project that was announced 2 weeks ago that we're really excited to launch, and we'll hope to present results sometime soon. And those are 5 years projects, so it will take time. So I also want to thank all the participants to this project, the members of my team, the members of the New York Genome Center that helped, the steering committee, and all of the different partners that we had for this project. Thank you very much for your attention, and I'm happy to answer any question.
Thank you. Very interesting talk. Any questions?
Great talk, Nicolas. Great talk. Actually, it's not a question. It's more like a comment. I think your study of the CDK cyclin D1 amplicons is very nice. You're using what, nanopore or?
So it's mostly an Illumina study where we did the cohort, I think 77 samples from 50 different patients in bladder cancer, and we did tumor normal whole genome sequencing with Illumina. What we discovered was an extrachromosomal DNA and so for those of you who don't know, there are multiple ways in which a gene can be amplified, and some genes are very highly amplified. And one way in which a gene can be amplified is through an extrachromosomal DNA, where a piece of DNA gets circularized and can be brought into multiple copies per cell. And there are various reasons why that's a nice way to increase diversity within a tumor. And so here we identify that the gene cyclin D1 was amplified as an ecDNA in bladder cancer. And in order to validate that it was an extrachromosomal DNA, we did long-read whole genome sequencing with Oxford Nanopore, which is not a perfect way to validate because the reads that you obtain from ONT are about 10 kb, whereas the ecDNA is about 1 megabase, right? So you have a lot more confidence in the ecDNA, but it's not a perfect experiment. We have FISH experiment that really validate that this amplification is really as an ecDNA in cells.
But it's an important concept, especially as a lot of people in genomics now don't have like a background in cytogenetics. So this was known in cytogenetics,
Absolutely.
but is not, there's many people in genomics who don't realize that because of the way that we present copy number changes across the genome, it looks as if the amplified gene is amplified at the chromosomal location, but in fact it pops out and actually also reintegrates, picks up another gene like you showed, FGFR1, I think.
Yes.
And so
on.
So
FGFR1 is next by cyclin D1.
No, FGF19 is.
So, and actually, I think we should mention, so those ecDNA were called double minute in cytogenetics and were heavily studied in Japan in the 70s and 80s. And so a lot of people that are really specialized on studying ecDNA are now going back to the papers and really identifying how most of it was known. And it took about maybe 10 or 15 years of 10 years of cancer genomics with next generation sequencing before people rediscovered those things. But really, it's a rediscovery. And they, for some reason, gave it a different name. And so we can debate if that's, that's the same thing as double minutes, but it was known. I think now we can study it in a lot more detail. We can look at somatic signatures on those ecDNA, for instance, right? So here, that's one of the things we do, like what are the processes operating on that ecDNA compared to the rest of the chromosomes? And so, yeah, there's a lot more work to be done on that.
It's really amazing scale, and I'm so overwhelmed by all the data. So I'm just curious. So do you have a bandwidth of considering the environmental factor? For example, immigrant has first, second, third generation, and so on, and histone modification, etc.
Yeah, that's absolutely the way to go. Very clearly, our project was focused on whole genome sequencing, on the type of things we know how to do, and the importance of that we perceive to have in the database. Right? In order to really work with those databases, it's better if those databases are more representative. We are not going to fully explain genetic disparities or health disparities by this type of work. We can, really start to explain, to quantify a small portion of it, but a lot more needs to be done. And that's really why this latest project that we announced has a very extensive collection of social information. Where did people live? What was their work? Where did they work? What type of exposure were they exposed to? As well as an exposomic component, and trying to have a mass spec blood sample with a mass spec experiment that could potentially inform on what type of exposure they were exposed to, right? Did they live in a polluted environment? The type of environmental factors. Definitely there is something to be said about epigenetic and about, histone modifications, methylation, all sort of other elements that can be collected and that will affect the way cancer is more or less aggressive, the way cancer responds to treatment, and all of that. We need more studies, we need more people, we need more data, we need more multidisciplinary studies, I think is really important in order to really make important progress.
Thank you very much. It's very interesting study. So I have two questions. One is related to the ecDNA. So we also observed ecDNA in gastric cancer, but I would like to know, in your cases, ecDNA happens in early stage or advanced cases? Because some studies say that it is in the precancerous regions, ECDN is already detected
Interesting.
in esophageal cancer.
So I think in our case, that's a separate part of the study. So only some of the patients we have enough samples to do the phylogenetic reconstruction and try to know what happened early on. I don't know if the samples in which we detected the ecDNA were part of this rapid autopsy, and so I don't know. I'm not even quite sure how we would know the timing of when it happens. We in the benign urothelium samples that we have, I think we did not detect an ecDNA. We are doing some other work on somatic mosaicism in healthy tissues, but yeah, we're looking at that. Like in any tissues, we are all every day accumulating mutations, right? And we don't all get cancer every day. But how frequently do we have ecDNA in healthy tissues as part of somatic mosaicism? I don't know, and I'm sure that's going to be established in the next few years. But yeah, it's possible. For this particular study in bladder cancer, I don't think we have the answer for that.
Okay.
Yes.
And the second question is about your Survive project. So it's very interesting because you have multi-omics or studies, including immuno decompositions. Are you going to do some single-cell analysis of samples?
Hopefully we will do some single-cell and some spatial profiling. Exactly which technology, I'm not quite sure yet. And that's really going to come in the later end of the project, right? Okay. We will really start with social determinant of health, germline genetic, and then progressively somatic, cancer sequencing, and then immune profiling.
Okay, thank you.
Thank you.
Thank you very much. I would like to ask you about the survey.
Yes.
And there are many multiomics data, but I would like to ask you about the exposome.
Yes.
And do you have some specially interested factors?
So, as you can imagine from what I described, my task in this project is the third one, the genomic part. Mm-hmm. We are partnering with a group at Columbia University that has a really specialized expertise on exposomic. And they've done that mostly in neurodegenerative disease, in Alzheimer and Parkinson, for instance. Right? I think it would be their first cancer project. But we are going to partner with them. So it's a little bit too early for me to answer the question. But it's definitely exciting to triangulate those three methodology and try to really quantify the effect of each other and build methods that combine the three type of information. And as I said, that's really the power of multidisciplinary approach is going to be to combine those three. So that's what I'm very excited about.
Thank you.
Thanks.
Any other questions?
I see some young future professors here. And any questions from young generation? No? Oh, okay, so I think we are running out of time, and so thank you very much for a nice talk. And